Right, welcome back to the continued portion of our lecture. We were on virus resistance in the previous portion that we just finished. So I'll start off from there itself. Virus resistance. Now, plant viruses often cause a considerable crop damage and significantly reduce yields. Therefore, in the absence of effective chemical treatments, plant breeders have attempted to transfer naturally occurring virus resistance genes from one plant strain cultivar to another. However, resistant cultivars often revert to virus sensitivity, and resistance to one virus does not necessarily account for resistance to other similar viruses. Natural virus resistance can be achieved in different ways, and viral transmission can be blocked, established of the virus can be prevented, or viral symptoms can be bypassed or resisted. Genetic engineering has been used to develop non-conventional types of virus-resistant transgenic plants. Before I can continue, I'd like to welcome all of you once again to daily lectures from New Age Cosmetic White EdTech Healthcare Consultancy, and I'm Dr. Bobby. For all the lectures that you've seen so far, please make screenshots, listen to them, make notes, and to the best of your capacity and capability. Kindly do not commercialize any aspects of these lectures or screenshots or the illustration shown to you exclusively for study purposes. And various lectures you could find links to certain videos. Those are only for references, so you get to have an idea of what's being taught. Right? So please use them to the best benefit of your studies, to all the students in distress and various inconveniences. Okay, and we'd like to thank all the authors and the publishers for the various texts and details that they've given us. Invaluable by all means. Thank you so much. Gratitude and courtesy to all of them. Continuing back with the text of the matter proper. Viral coat protein-mediated protection. Now, when transgenic plants express their gene for a coat protein, which usually is the most abundant protein of a virus particle, a virus that normally infects those plants, the ability of the virus to subsequently infect the plants and spread systematically is often greatly diminished. For a long time, the precise mechanism for by which the presence of coat protein genes inhibits viral proliferation was not understood. However, it is now thought that it likely works through the generation of RNAi. Moreover, the antiviral effect occurs early in the viral replication cycle, and as a result, prevents any significant amount of viral synthesis. This feature is an advantage because it decreases the probability of selecting for spontaneous viral mutants. And that can overcome this resistance and replicate in the presence of viral coat protein. The viral coat protein gene approach has been used to confer tolerance for a number of different plant viruses, as shown on table 19.2. With this approach, researchers have developed virus-resistant transgenic plants for a number of different crops. Although complete protection is not usually achieved, high levels of virus resistance have been reported. In addition, a coat protein gene from one virus sometimes provides tolerance for a broad spectrum of unrelated viruses. The utility of this strategy is supported by the observation that transgenic plants that encode viral coat proteins do as well in field trials as in the laboratory studies. In both eukaryotes and prokaryotes, an mRNA molecule that is complementary to a normal gene transcript mRNA is called antisense RNA. The mRNA, being translatable, is considered to be a sense RNA. The presence of antisense RNA can decrease the synthesis of the gene product by forming a duplex molecule with the normal sense mRNA, thereby preventing it from being translated, the antisense RNA to mRNA, thereby preventing all the I repeat, the antisense RNA mRNA duplex is also rapidly degraded, a response that is diminished or diminishes the amount of the particular mRNA in the cell. Theoretically, it should be possible to prevent plant viruses from excuse me, replicating and subsequently damaging plant tissues by creating transgenic plants that synthesize antisense RNA that is complementary to viral coat protein mRNA. One of many studies, the efficacies of the viral coat protein gene and antisense RNA approaches were compared by cloning the cDNA for the coat protein of cucumber mosaic virus CUMV into tobacco plants in two orientations, sense and antisense, one orientation per plant, and then testing transgenic plants for sensitivity to viral infection. The genome to CUMV consists of three separate single-strand pieces of RNA, each coding for a specific viral protein. In vivo, one of these pieces, RNA3, is processed to remove a portion of its sequence, thereby generating RNA4, which encodes a viral protein. To create transgenic plants that either produce normal mRNA and express the viral coat protein or produce its antisense RNA, the following steps were carried out. Isolation of RNA4, in vitro enzymatic conversion of RNA4 into a double-stranded cDNA, addition of linkers onto the DNA, insertion of the full-length cDNA sequences into cloning vectors in both orientations, in which each oriented sequence under the control of the 35S promoter sequence from cauliflower mosaic virus, the termination regulatory sequences from the plant gene for the small subunit of ribulose biphosphate carboxylase, formation of separate transgenic plants carrying the cDNA sequence in one of the two possible orientations. The triplasmid binary vector system was used to transfer both protein-producing sense and antisense RNA producing cDNA sequences to separate tobacco cells in the transgenic plants were regenerated as shown in figure 1912. The transgenic tobacco plants that express the CUMV coat protein were protected from viral particle accumulation and did not show symptoms of viral infection, regardless of whether the inoculum of the challenge protected transgenic plants only against low viral doses. Several groups of scientists have constructed transgenic plants that synthesize antisense RNA, copies of viral coat protein genes, and tested whether these plants can withstand a viral challenge. In all instances, the plants were protected against the invading virus only when low concentrations of the virus were used. At high concentrations, the plants were damaged by the virus. In addition, antisense RNA of viral coat protein genes generally afforded a much lower level of protection to transgenic plants than the sense versions of the viral coat protein genes, although the antisense RNA approach may not be an effective means of creating virus-resistant plants. It may be possible to use small interfering RNA, double-stranded RNAs, about 21 nucleotides long, to protect plants against invading viruses. In this case, the interfering RNA would act to target specific mRNAs, example mRNAs, encoding viral coat protein for nucleus digestion. And often, field crops are exposed to several different viruses, any one of which may damage the plants and lower the final yield. Ideally, transgenic plants should be resistant to more than one virus. With this in mind, T1 plasmid binary vectors expressing one or more coat protein genes for CUMV, zucchini, yellow mosaic virus, and watermelon mosaic virus 2 were used to transform yellow crookneck squash cucurbita pepper plants. Transgenic plants that contained the coat protein genes from all three viruses were resistant to damage by all three viruses under laboratory conditions. Initially, transgenic plants expressed coat protein genes for zucchini yellow mosaic virus and watermelon mosaic viruses. Mosaic virus 2 were tested under field conditions by using aphids, which are small insects that naturally transmit these viruses to developing plants. The transgenic plants that express both code protein genes were completely resistant to infection when the two viruses were transmitted at the same time. And transgenic plants that contain the protein gene of all three viruses were resistant to damage by all three viruses under laboratory conditions. All three viruses initially transgenic plants expressing code protein genes with zucchini yellow mosaic virus and watermelon mosaic 2 virus were tested under field conditions by using aphids, which are small insects that naturally transmit these viruses to developing plants. The transgenic plants that express both code protein genes were completely resistant to infection when the two viruses were transmitted at the same time. On the other hand, while transgenic plants expressing only one of the two viral code proteins were delayed in succumbing to the viruses in comparison with the non-transformed control plants, all of these plants eventually developed severe symptoms of viral disease, making them unfit for sale to consumers. More recently, transgenic squash plants that express viral code protein genes with zucchini yellow mosaic virus, watermelon mosaic virus 2, and CUMV have been tested in the field. Following demonstrations that they effectively protected plants against diseases caused by any or all of these viruses, these transgenic squash plants were made commercially available. The increase in plant yield resulting from virus and protection depends on which virus the plants are challenged with, how great the viral pressure is, and the time of the growing season. Despite these many variables, one study has estimated that transgenic squash with resistance to these three viruses subjected to severe viral pressure could produce as much as a 50-fold increase in marketable squash over non-transgenic varieties, clearly using more than one viral code protein gene is an effective strategy that should be useful in developing a range of transgenic plants that are resistant to all of the major viruses that normally inhibit their growth and development. However, it must be borne in mind that in order to satisfy the variety of consumer tastes, there are a large number of squash varieties, all of which are potentially susceptible to these viruses. In this approach, all of them would have to be genetically engineered in a similar manner. In addition, those transgenic lines that are resistant to three different viruses are also still susceptible to papaya ring spot virus type W, so complete viral protection of summer squash will require the introduction of the viral code protein for this virus as well. The phenomenon of using plant-coded viral gene to disrupt the virus life cycle and thereby confer resistance to the virus is sometimes called homology, dependent in gene silencing, formerly called co-suppression. In homology-dependent gene silencing, the addition of new copies of a gene to the genome inhibits expression of both the introduced gene and the previously present endogenous copies, or in the case of viral genes, those genes that are synthesized after infection. In fact, in some cases, the plant's own defense mechanisms may include the possibility of homology-dependent gene silencing. Perhaps the most successful employment of the viral code protein strategy to prevent damage from a plant virus is the use of the code protein of papaya ring spot virus to protect papaya plants against the virus. In fact, these transgenic papaya plants are widely credited with saving the Hawaiian papaya industry, as shown in box 19.3. Protection by expression of other genes. RNase 3 engineered resistance to plant viruses generally as a result of expressing a viral code protein or other viral gene in the transgenic plant is usually an effective strategy only against closely related viruses. Since there are a large number of different viruses that could potentially infect a crop, it would be advantageous if plants could be engineered to be resistant to a broad spectrum of viruses. To do this, a strain of wheat was engineered to express the E. coli gene RNC for ribonuclease RNase 3, an enzyme that cleaves only double-stranded RNA. Most plant viruses have double-stranded RNA, as the genetic material when tested. Transgenic plants that express the RNC gene were resistant to several different RNA plant viruses. Unfortunately, plants that expressed this gene were often stunted and did not develop normally. This was probably a result of the interaction between the plant RNA and the enzyme. To overcome this problem, a mutant of RNase-3 was used. The mutant enzyme was still able to bind stoichiometrically to double-stranded RNA, but it no longer cleaved this substrate. The mutant gene RNC70 was introduced into wheat under the control of a corn ubiquitin gene promoter by microprojectile bombardment. Transgenic plants that express mutant RNase-3 developed normally and exhibited a high level of resistance to infection by barley stripe mosaic virus. In this instance, binding of the mutant RNase-3 to replicating barley stripe mosaic virus prevented viral replication, in addition to being useful with RNA viruses. This approach should be an effective strategy for eliminating viroid infection of plants. Viroids are disease-causing agents that with a circular single-stranded RNA genome that contains double-stranded regions formed by intra-strand base pairing. Plant viroids are difficult to control because they do not encode any proteins. Therefore, the viroid nucleic acid must be targeted. Pokeweed antiviral protein. In addition to immunizing plants against damage from viruses of expressing viral proteins in the plant cells, protection can be conferred by antiviral plant properties. For example, pokeweed Phytolacca americana has 3 antiviral proteins in its cell wall. Pokeweed antiviral protein PAP, which is found in spring leaves, PAP2, which is found in summer leaves, and PAP-S, which appears in seeds. Although they are only 40% identical to protein level and antibodies directed against PAP do not react to PAP2. They employ similar modes of action. Both PAP and PAP2 are ribosome inactivating proteins that remove a specific adenine residue from the large ribosomal RNA. Of the 68 subunit of eukaryotic ribosomes, when pokeweed plants are infected with viruses, either PAP or PAP2 is synthesized. Depending on the season and the ribosomes in the infected cells are inactivated. Based on the mode of action, PAP and PAP2 are good candidates for developing transgenic plants that are resistant to a broad spectrum of plant viruses. After a cDNA encoding PAP was isolated, it was introduced under the transcription control of the 35S promoter. The tobacco and potato plants with binary tie plasmid vectors, transformants that expressed a high level of PAP greater than 10 nanograms per milligram protein that had stunted growth. And transformants that express a high level of PAP greater than 10 nanograms per milligram protein had a stunted and mottled appearance and were sterile. On the other hand, plants with a lower level of PAP, 1 to 5 nanograms per mg protein, were normal in appearance and fertile. Thus, above a certain level, PAP interferes with normal cellular functioning in transgenic plants. Expressing PAP, the major side effects, a major effect of the antiviral protein was to reduce the number of lesions in virus-infected plants. When transgenic tobacco and potato plants expressing low levels of PAP were challenged with either potato virus X or Y, they developed significantly fewer lesions on the leaves than non-transform control plants. Transgenic plants that contain the cDNA for PAP2 express this protein at a much higher level than that observed for PAP, up to 250 nanograms per mg protein. Plants with greater than 150 ng of PAP2 per milligram protein had chlorotic lesions, while those with 10 to 10 nanograms of PAP2 per milligram of protein were normal. Transgenic plants that expressed a lower level of PAP2 and were otherwise normal were resistant to tobacco mosaic virus, potato virus X, and the fungal pathogen Rhizoctonia solani. While this gene is highly effective in the laboratory, it remains to be seen how it will function under field conditions. And on to single-chain antibodies. One way to protect plants against viral infection is to engineer the plants to produce antibodies that are directed against the invading viruses. This was done with some success by expressing single-chain FV antibodies against tobacco mosaic virus and tobacco plants. However, as a consequence of the variability between code proteins from different viruses, the strategy is not useful for providing broad-range resistance against several different viruses. The majority of plant viruses are RNA viruses, and many of them contain positive stranded RNA as the genetic material. These viruses all code RNA-dependent RNA polymerases that are essential for their replication. Thus, a single-chain FV antibody that recognizes epitopes that are common to the RNA-dependent RNA polymerases from different viruses should be an effective means of inhibiting the replication of all these viruses, thereby making transgenic plants that express these single-chain FV antibodies resistant to these viruses. That is because the antibody fragment can bind to RNA polymerases and thereby block the activities. In addition, since even in virus-infected cells, RNA-dependent RNA polymerases are found in only low concentrations, a high level of antibody expression is not required. And two-phase display libraries were screened against purified denatured fragments of the RNA-dependent RNA polymerase of tomato bushy stunt virus. The three single-chain FV antibodies that display the highest affinity were isolated and characterized. cDNA coding DNA that encoding of these single-chain FV antibodies was expressed in Nicotiana benthamiana, a close relative of tobacco native to Australia. The resultant transgenic plants were significantly protected against tomato bushy stunt virus and cucumber necrosis virus, and was partially resistant to turnip quincke virus and red clover necrotic virus. In addition, the single-chain FV antibody directed against tomato bushy stunt virus bound to the RNA-dependent RNA polymerase of hepatitis C virus, which is much more distantly related. This work represents an important first step in using a simple and straightforward strategy to develop transgenic plants that are resistant to a wide range of different viruses. On to the next topic, called the microRNAs. Now, one approach to developing plants that are resistant to a range of different viruses might include engineered plants that produce microRNAs or miRNAs that interfere with the viral replication by targeting the viral NRA or the viral mRNA for degradation. A recent series of experiments, starting with a 273 nucleotide precursor of a naturally occurring plant miRNA, scientists use PCR, that's polymerase chain reaction, to replace a small portion of the existing sequence so that the precursor could be processed to yield a 20 to 24 nucleotide long miRNA that was complementary to viral RNA. The newly synthesized artificial miRNA became part of an RNA-induced silencing complex in which the viral mRNA was specifically bound and cleaved. It is also possible to clone two or more different pre-miRNAs in tandem. This was done using turnip yellow mosaic virus and the turnip mosaic virus, so that plants transformed with this construct become resistant to two separate viruses. This approach can be made even more effective by targeting more than one portion of each viral RNA using several pre-miRNAs. Despite its intriguing possibilities, the system still requires a considerable amount of development before it is shown to be effective under field conditions. Herbicide resistance. A significant fraction of global crop production is lost through weed infestation every year. Despite the expenditure of $10 billion on more than 100 different chemical herbicides. In addition, many herbicides do not discriminate weeds from crop plants. Others must be applied early before the weeds. take hold and some persist in the environment. The creation of herbicide-resistant crop plants is one way to overcome some of these drawbacks. A number of different biological manipulations that will cause a crop plant to be herbicide-resistant can be envisioned. Inhibit uptake of the herbicide, overproduce the herbicide-sensitive target protein so that enough of it remains available for its cellular functions despite the presence of the herbicide. Introduce a bacterial or fungal gene that produces a protein that is not sensitive to the herbicide but performs the same function as the plant herbicide-sensitive protein. Reduce the ability of a herbicide-sensitive target protein to bind to a herbicide and to a plant with the capability to metabolically inactivate the herbicide. A number of these strategies have been implemented to produce herbicide-resistant transgenic plants. This approach has been so successful that more than 75% of the transgenic crops that are currently planted worldwide have been engineered to be herbicide-resistant, by far the most widely used. Herbicide is glyphosate, which is considered to be safe, cheap, effective, and environmentally friendly because it is readily degraded for non-toxic compounds in the soil. Glyphosate, trademarked as Roundup by the Monsanto Corporation, inhibits a key enzyme in the shikimate pathway, 5-enol pyruvyl shikimate 3-phosphate synthase EPSPS, that plays an important role in the synthesis of aromatic amino acids in both bacteria and plants. Plants resistant to this herbicide have been developed by putting in an EPSPS encoding gene from a glyphosate resistant strain of E. coli under the control of plant promoter and transcription, termination, polyadenylation sequences, and cloning the construct into plant cells. Transgenic soybean, corn, canola, tobacco, petunia, tomato, potato, and cotton plants that produce an amount of the resistant E. coli EPSPS sufficient to replace the inhibited plant enzyme are resistant to the effects of glyphosate. Thus, in these cases, the crop plant that would not be affected by glyphosate treatment, whereas the weeds would be. Crops that have been engineered to be resistant to glyphosate by this approach are said to be Roundup ready. Notwithstanding the many years of successful use of glyphosate and Roundup ready plants, two important factors are now changing people's thinking about this approach. Well, this is the fourth edition, so there will be a lot of changes by now. Please update your knowledge on whatever I've been speaking about right now. Right? In the first instance, the herbicide patent has now expired, and other companies are very active in the development of plants that are resistant to glyphosate using the other approaches. Secondly, there is a generation that worldwide agriculture has become too dependent upon a single herbicide, and that alternative strategies need to be developed. To find an enzyme that can inactivate glyphosate, one group or researchers say a collection of several hundred Bacillus sp strains for the ability to acetylate the glyphosate. The assay was based on the ability to measure N-acetyl glyphosate and the supernatant of permeabilized cells. The three strains, all Bacillus licheniforms, that had the highest level of glyphosate N-acetyl transferase activity were isolated, and the enzymes were characterized. In all cases, these strains exhibited only a very low level of activity. Subsequently, the genes from each of these selected strains were encoding glyphosate and acetyl transferase activity were isolated. These genes were then shuffled, see Chapter 8, numerous times, each time selecting the strain with glyphosate and acetyl transferase with the highest level of activity. Now, the following 11 iterations of DNA shuffling, the enzyme catalytic activity or efficiency improved by about 10,000-fold. Interestingly, the modified enzyme still functions as a glyphosate and acetyl transferase. After so many rounds of modification, the amino acid sequence of the modified enzyme is only 76 to 79% identical to the amino acid sequence of the parental enzymes. Finally, the modified glyphosate and acetyl transferase gene was introduced into Arabidopsis, tobacco, and corn plants. The transgenic plants, which express the enzyme in the plant cytosol and both morphologically normal and fertile. This lecture will be over in a couple of seconds. I'll see you in the next lecture. So, thank you.